that I have called biological control agents against sunflower pathogens. I will go through 4 points: the current situation of the biological control of sunflower diseases, biological control by means of entomopathogenic fungi, biological control by means of soil-inhabiting bacteria, and sunflower. Biological control is one of the tools we have to manage crop diseases, or sunflower diseases in this case, together with agronomical, physical and cultural methods, chemical control, or genetic resistance. Biological control is the use of living organisms to suppress the population density or impact of specific pest organisms, making it less abundant or less damaging than it would otherwise be. And it's one of the tools for integrated pest management. Biological control agents are focused on the fight against invertebrate pests. In this case, we use predators, parasitoids, pathogens. We can also fight against weeds using herbivores or pathogens. And our case now is the biological control of plant pathogens using antagonistic microorganisms or inducers of plant resistance. In sunflower, some works on biological control of diseases started in the 90s of the last century, and they were focused on the identification of microorganisms from the sunflower rhizosphere, and they deserve attention because they have a putative antagonistic effect against the diseases, and also they are good colonizers of the plant roots. There were some works with Pseudomonas and Bacillus that were able to control Macrophomina fasciolina and Sclerotinia grossly. Even Pseudomonas putrida showed some reduction of sunflower downy mildew under greenhouse conditions, but these works didn't go further. Later, some plant extracts have shown an in vitro effect against some soil-borne fungi affecting sunflower. And this is the case of some extracts of Lycium europaeum having high content of chlorogenic acid. They inhibited the mycelial growth of Verticillium dali and also of Sclerotinia sclerotiorum, and as well as the formation of Conidia bat Verticillium or Sclerosia bat sclerotinia. And a recent work is focused on the interest that Trichoderma arsianum has on the inhibition of mycelial growth of sunflower pathogens, but they are seed-borne, not soil-borne, like Alternaria alternata, Bipolaris sinodantis, Fusarium pulmorum, and Fusarium oxysplum. So, sorry. All this makes us think that new approaches and new options and further research can still be explored for an effective, if not complete, at least partial control of sunflower diseases using biological options. So going to the use of entomopathogenic fungi, entomopathogens are fungal entomopathogens are natural enemies of pests, particularly phytophagous insects, and they are highly efficient. They are present in different environments and are subjected to active commercial development. These fungal entomopathogens have 2 ways of action. One of them is by contact. They can infect the phytophagous insect through the tegment. They cause the death. The fungus sporulate on the cadaver, and then finally the spores are released to the environment. And the second way of action is the endophytically way of affecting the pest. The entomopathogen infects or colonizes the plant, then the phytophagous insect feeds from that plant and becomes sick and dies, and again the fungus is able to spread the conidia to the environment from the body of the insect. We use the approach of the entomopathogenic fungi against the studying the effect against verticillium wilt of sunflower, a very distributed disease with more than 2,000 host plant species, known because it's able of surviving in the soil for long periods of time in the form of microsclerosia. The symptoms in the plants are intervinal chlorosis, yellowing of leaves, progressing upwards, also stem lesions, and there in the corner you can see the morphology of the fungus in a culture media. The second disease we were working on was the sunflower downy mildew caused by Plasmopara abstedi. Very important, also distributed worldwide, caused by an obligate parasite, able of infecting the plant by two ways, through primary infections from resting spores surviving in the soil and affecting seeds and seedlings, or secondary infections from soil spores that infect leaves and they have low economic impact. The symptoms of the disease are pre- and post-emergence damping off, chlorosis, bordering the main leaves, the main veins of the leaves, and progressing downwards, and stunting of the plants. And as in the case of verticillium, genetic resistance is one of the control options we have. Also in the case of downy mildew, there have been chemical alternatives, and currently oxatiapiprolin is the active ingredient approved in most of countries in Europe. So the objectives were to determine whether the strains of different entomopathogenic fungi species were able or were suitable candidates as biological control agents against verticillium. We did in vitro experiments, also in plant experiments in the greenhouse. We assessed the time lapse of the entomopathogens in the substrate and conducted a molecular detection of the microorganisms in the plants. Then the second objective was to determine the effectiveness of entomopathogens, same strains of that we used against verticillium dahlia. We tested against sunflower downy mildew, against the disease on one side, and on the other side against by their effect on the direct effect on the plant. And we determined if they were colonizing the plants. So in the case of the studies with verticillium, we did dual cultures. We used strains belonging to two different species, Metarrhizium bruneum and Boveria bassiana. They are very known as entomopathogens. Against 6 different isolates of verticillium, we did dual cultures in medium, and we assessed the inhibition of mycelial growth by comparison with the growth of the control. And then we also incubated some slides under the media between two colonies, and we observed the growth under the microscope. The results we saw, the inhibition of mycelial growth was between 8 and 63%, depending on the entomopathogen and on the isolate of verticillium we were working with. And the most efficient strain of entomopathogen was one of Boveria bassiana, and the less efficient one belonging to Metarrhizium bruneum. Concerning the antagonists, in most of the case we observed mycelial overgrowth and antibiosis in one of the strains of entomopathogens. Now, this is the experiment in the greenhouse. We were using 4 strains of entomopathogenic fungi, one isolate of verticillium dahlia against in one susceptible genotype. First, we treated the soil with the entomopathogen, then we sow the seed in that pot, And after 30 days, we extracted we uprooted the plant, and we inoculated it with the verticillium. And the plants were incubated in the greenhouse between 1 and 2 months. We observed the disease progression weekly. We calculated the area under the disease progress curve, and we also did an assessment, a weekly assessment of the populations of the pathogens in the soil. Here, in the upper graph, you see the disease, the severity of disease. In the other graph below, I'm showing the area under the disease progress curve. The results are very similar, and the most complete ones are those belonging to the lower graph. And we found that two of the strains were significantly different than the control. One of them was a Metarhizium brunum, and the other one of the Boveria bassiana. When we analyzed the populations in the soil, we found that here we can see in the first week after the inoculation, we had 20% of symptom severity. By the fourth week, we had 62% of symptom severity. Weekly, we had populations in the soil in nice levels. Only one of them had a strong increase the last week. So we saw that the entomopathogens were able to persist efficiently in the soil during the experiment. And we did the molecular analysis. Here, there are the DNA, the lengths of DNA. This is sunflower, verticillium, entomopathogens, and these are the plants with no verticillium, only entomopathogens and sunflower. This is the DNA, and treated plants in both cases. So where the sunflowers were inoculated with verticillium thallium, we detected the pathogen, but not the entomopathogen by molecular methods. Here, this is verticillium together with sunflower. Here we have entomopathogens together with the sunflower, but here we don't have them together. So what we conclude here is that the protection that the entomopathogens Boveria bassiana and Metarhizium brunum conferred against verticillium wilt might not be plant located, but it's more likely to be the consequence of the competition with verticillium thallium in the soil, and this is why they do not penetrate the plant at the same time. So in the case of downy mildew, we did a similar approach, experimental approach. We used the collection of strains against one isolate of Plasmopara steady and one susceptible genotype. We germinated the seeds and sowed them in Holland and Knox culture medium in glass tubes. First, we treated the seeds with the entomopathogens, and then 24 hours later, we added the pathogen. And we observed the sporulation between 1 week and 10 days later. We didn't find any difference between the treated plants and the control. Even some of them had higher levels, but as we used transform values for the statistical analysis, that's why there are no significant differences here. And we didn't find any difference on severity, and we didn't find any difference on shoot height and root length or dry weight of shoots and root size. We did microscopical detection of we evaluate the effect of the entomopathogens in the absence of Plasmopara steady, but only in the plant. And we found that some of them reduced some of the growth variables of sunflower, particularly this one. All the values were significantly lower, and some others had an effect, a detrimental effect on the roots. When we did the microbiological isolation inside from the tissues of the plant. We did a plating on Saburat dextrose agar from tissues of roots, shoots, and leaf tissue, and we saw that none of the entomopathogens was recorded from plants inoculated with Plasmopara steady, as we already knew from the previous results. And when the entomopathogens were alone in the plant, they were isolated, also with in percentages that varied depending on the plant part, being root, shoot, or leaf, and the strain of entomopathogen. This is the molecular detection. In each of the groups, we have 4 treated plants, the DNA from the entomopathogen, the DNA from sunflower, and we were only able to identify the pathogen in some of the treatments in all the plants, and only in some of the plants in other treatments. Never the Plasmopara steady. So DNA from the entomopathogen was only amplified in plant samples treated with entomopathogens, but not inoculated with Plasmopara steady. So what we can conclude from this part of the work is that the obligate nature of Plasmopara steady infections and the strong dependence of Metarhizium and Boveria species on soil ecology and characteristics seem to condition the bioactivity of entomopathogens against down mildew. So this bioactivity is highly dependent on environmental conditions involving the experimental soil system that was the one we used. The third point is biological control by means of soil-inhabiting bacteria. The biological control using soil-inhabiting microbes has been a successful strategy against some soil-borne pathogens, and it has been widely used in the management of Fusarium and verticillium diseases. The most common or known genera with effect against this type of diseases are Pseudomonas, Bacillus, Streptomyces, Trichoderma, non-pathogenic Fusarium historum, and some muscular mycorrhizal fungi. In our group, we have identified some biocontrol agents that are effective against verticillium wilt of olive tree and Fusarium wilt of vanilla. Some of these come from the rhizosphere of the plants and the roots and are native constituents of the root, endophyte, what we call the endophyton. And then one of them, Pseudomonas simiae, is highly known, and we have used it a lot because it has very interesting biocontrol and plant growth promoting abilities and displays significant biocontrol of verticillium wilt of olive and Fusarium wilt of banana of both diseases. Again, we assess the effect of this bacteria against verticillium wilt, and in this case against sunflower broom rape caused by Orbanche cumana, a very known holoparasitic plant that depends entirely on sunflower for completing its life cycle, has a very strong impact on the production. Initial symptoms appear at the beginning of flowering of the crop, and by the time flowering is full, the stems emerge above the soil. The main way of controlling sunflower broom rape is genetic resistance. Also, different races are identified. It's a disease that is located in all Eurasia. Here I have marked all the countries where broom rape has been identified, and in orange are highlighted the affected areas. So the objective was evaluating the in vitro antagonism of soil inhabiting bacteria having good effects against verticillium wilt of olive and banana, wilts of olive and banana, in this case against verticillium dahliae affecting sunflower. And also we evaluated under greenhouse condition the effect of this bacteria on Orobanche-Cook-Mann infection of sunflower and also on the growth of sunflower by itself. Some preliminary results suggested that were presented in 2018 suggested that the bacterization of sunflower seeds with some of these bacteria, some particular strains of these bacteria could promote plant growth. This is a collection of bacteria, the reference taxa host of isolation being banana or olive, previous works, and experiment were repeated. Plates in the case of verticillium dahliae, greenhouse in the case of Orobanche-Cook-Mann. Well, the picture here is visible. I cannot see it here, anything here. This is the aspect of the isolate of verticillium. One of the isolates of verticillium we used. Actually, we used two of them, one from Andalusia, Spain, the other one from Turkey, against 10 bacterial strains. Like before, we confronted two colonies in different nutrient media and calculated the inhibition of mycelial growth. So the results show that we found, first of all, we found these significant differences of inhibition between isolates of verticillium dahliae. So we did independent analysis for each of the isolates. 5 bacterial strains showed interesting results, meaning more than 70% of inhibition against both of the isolates, and 2 additional strains inhibited only one isolate. When we went to the greenhouse, we included 10 strains belonging to Bacillus, Penibacillus, and Pseudomonas species in genera, 1 population of Orobanche-Cook-Mann, and 1 susceptible confectionery sunflower genotype. The experiment was in the greenhouse for 5 weeks and this temperature. And we observed the growth of the plants, the height of the plants, weight of roots and above ground plant pads, and counted the number of nodules in the roots. These are the results for the control of bloom rate. What we saw is that all the bacteria significantly increased the weight of the roots, and some of them also caused lower number of nodules. And some of them, 5 of them, were associated to taller plants. Now on growth promotion, all except one of the bacteria significantly increased the weight of the roots, all of them except this one. Only one, there were no significant differences on the weight of above ground pads, and only one of them negatively affected the height of the plants. So as conclusions, we can say that the protection that Bovea bassiana, and from the first part, okay, the part with the entomopathogens, the protection that Bovea bassiana and Metarrhizium brunnum confer against verticillium wilt might not be plant located, but is more likely to be the consequence of the competition with verticillium dahliae in the soil. The second one concerning downy mildew is that the efficacy of pathogens against Plasmopara steri might be related not to environmental conditions we use in our works. This is not unexpected, since Plasmopara steri is an obligate parasite, that shelters into the plant, and Metarrhizium and Boveria are soil dependent and highly affected by soil ecology and characteristics. And concerning the part with rhizobacteria, although the inhibition of verticillium by soil inhabiting bacteria dependent on the fungal isolate, five strains of bacteria showed promising results. So future research will address or should address the study of their efficacy under greenhouse conditions. And the last one is that the bacteria were associated to increases of root weight of sunflower, irrespective of the presence of Orobanche cumana or not. And in plants inoculated with Orobanche cumana, some of the tested strains were associated to low number of nodules. And this is all. Just this is my former PhD student who did all the part with entomopathogens. He had his thesis defended last year because this work comes from long ago. And these are our collaborators on the part of soil inhabiting bacteria. Thank you very much.